My name is Nick. I'm a CEO and co-founder of ASIMICA. We all know microbes and we know stem cells, but you may not realize that microbes do not form stem cells. During my PhD, I made an innovation of microbial stem cells. This invention opens new avenues in the market of microbial fermentation technologies. I was always fascinated by the power of fermentation as a new way of manufacturing of goods for people. It uses cells as factories grown in bioreactors. Genetic engineering enables the cells to produce products of the future, pharmaceuticals and fuels, artificial foods and new materials, and many, many more. It is a massive market, over 150 billion now, expected to double by 2025. But despite all advances in science, the real green revolution in production is still largely a dream. So why is that? Biofactories, unlike traditional factories, cannot operate non-stop. Every few days, the production has to be shut down. Each restart of the process is time-consuming and expensive, leading to 20 to 40% downtime lost. And this is limiting economic efficiency of biomanufacturing. The reason is that microbes inside bioreactors are suffering from the stress of production. They've been genetically engineered to make protein products, but like any worker, they break down and get old, and they accumulate mutations. It gets to a point where they cannot produce anymore. So many biotech companies are aiming to extend production lifetime in their bioreactors. A lot of genomic tricks were designed into microbes to make them stronger, more productive, more resistant to toxicity and mutations. But while you can keep your workers more fit, you cannot make them young again. They are getting old. It's genetics. What I'm going to tell you about today is an ultimate way to help this situation. At ASIMICA, we invented a method to repopulate biofactory with new workers during the process. We are doing it using our innovation of microbial stem cells. Much like human stem cells, which keep our tissues functional for decades, microbial stem cells are constantly replenishing the fraction of young and productive microbes inside bioreactor. But I said before that microbial stem cells do not exist, or they did not exist before ASIMICA. We know from school that microbes divide into two identical twins. During my PhD, I found a way to make this cell division asymmetric. That explains the name of our company. In this image, you can see asymmetrically divided cells, marked with red, generating new green young factory cells. Red cells have stem cell properties. Microbial stem cell technology is a new idea, and ASIMICA is the first company that designed its principles. And we are protecting our innovation. Our non-provisional patent was published a year ago. I published scientific principles of this work in the Nature Chemical Biology journal. The follow-up publication on mathematical modeling of our systems is in press. Our models suggest the highest impact of our technology for production of molecules which are highly toxic for the host cells and are most challenging for the industry. During our time in IndieBio, we have experimentally simulated this situation. We are making a protein called GP61. It's used in poultry and nutrition fields, and it's toxic for very bacteria which are making it. In our trials, we observed that in conventional cultures, after 20 hours of production course, majority of cells were broken down like exploded. At the same time, in presence of microbial stem cells, the culture was still healthy and producing. In subsequent 20 hours of production, conventional cultures almost stopped making any product, so the cells lost their potential for production. In presence of stem cells, the culture was still actively making a product. It is an early indication of the potential impact of our technology. And we are testing it on production of other industrial products. That includes jojoba oil, a valuable product in the cosmetics market. You can see yellow cells in the left photo which are making it. We are also making insulin, because it's such a huge market in the pharma industry. Other trials we have are with enzymes used in the food industry. Chymosin is necessary for making cheese from milk. Xylosazomerase is used for making high fructose corn syrup. And here we also observe significantly high protein yields using our method. We are approaching our future customers. We are already in the early stage partnership with a large German company which is using E. coli for production of drugs and food ingredients. A dozen of other companies are waiting for our scientific updates mainly on scaling up our experiments and transferring our technology to other fermentation platforms. In these partnerships, we are a B2B strain engineering company. We can enable strains of our partners with stem cell properties, increasing their productivity. We are asking partners for a flexible % royalty of top line of their production, in return boosting their profits. Imagine a company making 100 million revenue from one product. Half of it can be a cost of production. Our service can decrease the cost of production and increase their margins, and the royalties for ASIMICA would be calculated out of the delta we provide. We expect to become revenue positive in 2 years from now. In the next 9 months, we'll continue optimizing our system, scale up our experiments, and execute trial contracts. Once we de-risk our science, we'll be raising a seed round to hire more strain engineers and fermentation scientists to speed up transferring of our technology to other fermentation platforms, bacteria, yeast, and filamentous fungi. I got my education and early career experience back in St. Petersburg, Russia. Then I moved to Wyoming to do PhD in Dr. Grant Bowman's lab. That ended up launching a company. Dr. Grant Bowman is a well-recognized scientist in the field of microbiology. Jacob Guidry was in our lab for over 6 years, starting as an undergraduate fellow, continuing as a PhD student, and then joining ASIMICA. We also have support from a board of advisors, including Dr. Nathan Hilson from Joint Bioenergy Institute, Dr. Andrila Mukhopadhyay from Berkeley National Lab, Dr. Mark Gamelsky, who is actually co-inventor of microbial stem cells, and Todd Slaby, a consultant who helped us to connect with industrial partners. We are welcoming everyone interested to join us in this exciting journey of creating a new age of microbial fermentation.